So, yeah. So today we are very excited to have Dr. Lauren Alteio with us to talk about her work on soil microbial communities and microbial interactions. Dr. Alteio is a UMass Organismic and Evolutionary Biology alumni. She completed her PhD at UMass in 2019, where she worked on soil microbial communities using sequencing. And since 2019, she has been at the University of Vienna in Austria, also working on understanding microbial interactions in breakdown of carbon in soils. While she was at UMass, she worked with Life Science Cafe to plan and host some of these cafes. And she also gave a cafe on her PhD work. So yeah, we are very glad to have her this evening to talk about her work. Before we get into the discussion, we will have a short brainstorming exercise. So you have the link in the chat. For those that are unfamiliar with it, it is a word cloud, and we'll be using the Mentimeter website. So the question, we want to have an idea of where we are all coming from in terms of this topic we will be having the discussions on. And the question that we have is, what comes into mind when you hear of bacteria? So what is the first thing that comes into your mind when you hear of bacteria? So look in the chat and follow the link and let us see your thoughts. Good. Yeah. So what is the first thing that comes into mind? And we'll give folks a few minutes to let us know their thoughts. How great. Okay. We have constantly evolving, fascinating, diverse, soil, everywhere, ecosystem. That's awesome. Yeah. 16S RNA. Great. Diversity. Beautiful. Awesome. Yeah, those were all great thoughts. Mysterious. Yes. Yes. What are they doing in the soil? Why are they here? Good bacteria or bad? Oh, our grandparents. I like that. I like that one. So, yeah, cool. Tiny, symbiosis. Great. Things that run the world. Yes. Yeah, decomposition. Wow, these are adaptation. Extremophils. Awesome. Beginnings. Yeah, these are all wonderful, thoughts. Yeah, that's great. Thank you all for participating in the quick exercise. So we will jump into our discussion with Lauren. So Lauren, welcome.
Thank you all for inviting me. It's really nice to be back and part of Science Cafe. So I really appreciate the invitation. And judging from this word cloud, it looks like we have some microbial ecologists or community ecologists in the audience, so that's really nice that you guys are thinking about microbes.
Awesome. So to start with, for someone out there that is wondering what are even microbes and what are these organisms up to in the soil? So I'd like to ask, what are microbes and what are they really up to?
Yeah, so I can share, actually, I have a few slides just for
Awesome.
visual. Hold on one second. I'm going to share my screen. Okay. Sorry, can you see the presentation?
Yes.
Okay, so just to get a picture of what we're talking about here. So the word microbe encompasses a lot of different organisms. So it includes bacteria, which are microorganisms. Then there are fungi, which are multicellular organisms, as well as viruses, which we well know, infect us, but bacteria have their own viruses, as do some of these multicellular organisms. And then we also have microbial eukaryotes like amoeba that live in the soil. And so all of these organisms are below our feet and are under this umbrella of microbes.
Great. Okay. So let's also talk about microbial diversity. Could you briefly expand on what microbial diversity is?
Yeah, so as scientists, we're just beginning to scratch the surface of what the diversity actually is. And so when we think of diversity, we think of how many species there are, for example, and this may be easier to think about when we think about species of mammals, for example, or species of plants, where we can go out and really count the organisms that we see. But for microorganisms, because they're so small, it's difficult to really assess what's out there. So we're just trying to understand how many organisms there are, and it's a complicated question.
Yeah, awesome. So knowing that there is so much biological diversity in the soil, why is that so? And what are the factors that could be potentially driving the biological diversity within the soil?
Yeah, so this is also a huge area of research, but I think that a lot of the diversity is generated and maintained because of how complex soil is, for example. So I can skip ahead to like here. So we have a really complex structure of soil. So if you imagine we take a core of soil, which is probably a few tablespoons worth of soil, and within that there are millions of species, and soil has kind of this hierarchical structure where we think of having progressively smaller and smaller aggregates or clumps of soil. And so this gives microorganisms places to hide, and it also gives them access to certain types of nutrients that other microorganisms may not have access to. And so in that way, they can kind of diversify in their own little pockets of soil.
Oh, good. So we have seen you work on soil microbes over the years. Can you share with us why you are so interested in soil microbes and why at all should we care about them?
So I like that someone brought up our grandparents in this word cloud, because I think I initially became interested in science because of all the time I spent in my grandparents' garden. And so my first connection to soil and to ecology even was planting things with my grandparents. And so why should we care about microbes? I personally find them fascinating because you can't see them. And it's like this whole secret world that runs the world that we can actually see. So they're responsible for not only breaking down leaf material or carcasses of animals that might end up in the wild, but they also provide nutrients to the plants that we need to grow to keep ourselves alive. So they're really essential for life on this planet.
Great. I like that you already mentioned some of the importance, the roles they play in the ecosystem. And that brings me to the next question, which is the fact that I know that every species within the ecosystem is important one way or the other because of how they help in maintaining the ecosystem function and stability. But to be more specific, what role do microbes play in the ecosystem? And also how does microbial diversity within the soil help to sustain plant life?
Mm-hmm. So this is also a super complex question because microorganisms have many different roles. So some of them might fix nitrogen and make it available to plants so that they can take it up. Some of them might break down leaf material so that we don't have a huge amount of decay or of material just piling up all over the world. Others like fungi, they provide ways for other organisms to move around. So there's this whole fungal highway hypothesis where bacteria kind of skate along fungal hyphae throughout the whole soil system. In that way, they just kind of make nutrients available to plants and keep our ecosystems running.
Great. So we need them. And I think it also adds to the reason why we should care about them. So great. Okay. Another question that I would like to ask is, how do microbial communities change over space? And how do they also change over time in relation to like seasonality and some other factors like that?
Yeah. So I kind of tried to approach this a little bit with this figure here. We have the different spatial scales. So we have even like the ecosystem scale where we're taking like a whole soil core. Then we go down to like the aggregate scale. So we have a bunch of aggregates all together where we have water filling the spaces between. And then we have the within aggregate scale. And so, microbes are within and throughout this whole system. And so they experience different nutrient availabilities at different times. Like you mentioned, seasonality. So for example, in the spring, when the trees are just kind of waking up again and producing, like jump-starting their metabolism, they're also kind of exuding and letting out like carbon compounds that the microbes can take up. And so this also jump-starts the microbial metabolism. And so they go through annual seasonal cycles, just like the plants do. And maybe even on shorter timescales that we don't yet understand. So.
Yeah, good. I mean, they always say that change is a constant thing. So it makes sense that they are also changing in the ecosystem. So what is the social life of microbes? Could you briefly also tell us what that term at all means?
Yeah, so I also have some really cool pictures, I think. So these are from some papers from other scientists. But so the social life of microbes refers to the fact that they don't live in isolation, so that organisms so of the same kind can live within populations, and organisms of different kinds can live in larger communities. And within those communities, they need to communicate with each other, and they also share resources. So that goes into this whole public goods idea, where some organisms produce enzymes for breaking down certain things, and other organisms benefit from the breakdown of those compounds. And so in that way, they can live closely with each other. And there's also the flip side of that, where they're competing with each other for access to different things. And so as a result of this cooperation and competition, they sometimes form or often form spatial structures in the environment. And so you can get these kind of like webby looking structures or patchy structures, or these were actually on the bottom right-hand side taken from the tooth plaque of a human being. And so you can see different microbes showing up in different colors here in a reproducible way. So they form these kinds of structures. So we're trying to understand why that is.
Yeah, great. So many organisms in the ecosystem like have their own life cycles. Could you take us through a little bit the life cycle that microbes go through, like in the soil?
Mm-hmm. Yeah, this is also really complicated. I think we don't yet understand how this all works, but essentially organisms go through a process of obtaining carbon. So they take up carbon, they use that carbon to grow and divide and create new cells. And then they go through the process of death, and then the dead cells are broken down and used as a carbon source for other cells. So that would be a very broad overview of what's going on in terms of just the microbes in their life cycle.
Great. Okay, so we also know how our environment is always undergoing, anthropogenic disturbances. Mm-hmm. And how does the natural environment, how can we compare the natural environment with the disturbed environment as far as how the soil microbes go?
Mm-hmm. My first inclination is to say that the undisturbed environment might be more diverse because we're not introducing any disturbance. So this is also a huge area of research where people are looking into specific disturbances, like for example, adding a nitrogen fertilizer, and this changes the community composition of who's actually there. So I can't say for sure which microbes are changing, or if there are key microbes that change under specific conditions, but we're trying to figure that out still.
Great, yeah. So you mentioned diversity, that, the natural environment may be more diverse. How would you measure microbial diversity?
Yeah, so there are multiple ways that you can study microbes and microbial communities. So some of the ways include using cultivation, and this is when you think of putting microbes onto a petri plate and seeing, or into a liquid culture and seeing how they grow. But this is really complicated for soil organisms because we usually grow them by themselves, and they kind of, as we see in these pictures here, they like to grow with other organisms. So an alternative to that is using sequencing. And so that's what a lot of people do now, myself included, where we take a soil sample or a sample from the human gut, and we figure out which microbes are in there by sequencing them, by taking their DNA. And so, yeah, I guess diversity could be just classified as the number of species, which I'll use that term lightly, or someone mentioned the 16S ribosomal RNA gene. So this is like kind of a fingerprint of bacteria that we can use to tell who is who. Yeah.
Okay, so we already discussed how soil microbes may be changing over space and time, and you very much explained the impact of seasonality as well. And that makes me also want to ask how climate change could also be changing soil microbes and how these microbes behave. And climate change is like a broad topic everywhere that is being talked about these days. So how is that also potentially driving a change in soil microbes?
Yeah, so there's a large hypothesis out there that because microbes are really heavily involved in the breakdown of carbon within soils, as the climate gets warmer, the activity of the microbes might accelerate. And so they're going to start breaking down carbon faster and faster. And a product, I'm sorry, my dog is running through my apartment.
Yeah.
A product of that activity is an increase in carbon dioxide. And so that will increase further the amount of climate change that we're experiencing. So microbes could be really key in understanding what's going to happen in the future in terms of climate change.
Yeah, awesome. Thank you for all the wonderful answers, and you go through just clearly explaining all these terms and complexity regarding soil microbes. At this juncture, we will pause and take questions from our audience. So if you have a question, please click on the Q&A at the bottom of your screen and type it out. If you would like to ask your question out loud, you can also unmute yourself. You can raise your hand, rather. Raise your hand, and then you can unmute yourself to ask your question. So, yeah. Okay, so the first question from the audience is, have you studied how antibiotics change microbial soil diversity?
Yeah, this is also a huge area of research that I don't have anything to do with. But I find it really interesting that microbes from soil, that soils are where we get most of our antibiotics from. And so I think that microbes already have their defense mechanisms and ways to get around antibiotics in soil. And so I think it's really interesting, but I can't really answer the question, sorry.
Cool. And this question is also coming from Dee, and she is asking, how can you tell if your garden soil has good diversity of microbes?
Mm-hmm. Yeah, I guess my first response would be to get someone to sequence it for you, or maybe in the future we can develop some type of method that you can quickly tell who's there, but we're not there yet. Yeah, so I think sequencing is the best way to see the entire diversity of who's in your soil.
Good, thank you. And the last question, oh, not the last, the next question, maybe last, from Julia, and she's asking, what is EPS? I think you use those abbreviation. So what is EPS and
Mhm.
HGT?
So I think you're referring to here.
Yes.
So EPS, which is here, so this is a picture of a biofilm. So this is what microorganisms, so bacteria, use to stick to different particles in the soil, or in general. And so EPS stands for extracellular polysaccharides. And so this is a kind of a sugar substance. It also has other proteins and DNA suspended in it, that they use to stick. It's kind of a glue. And horizontal gene transfer, that's what HGT stands for, is a mechanism that bacteria use to exchange their DNA with one another. So it's an important way that bacteria generate more diversity.
Good. So the next question from Rachel. And Rachel wants to know if you have done any work with microbial interactions, specifically growing microbes in close proximity. Any method suggestions?
Yeah, so I'm trying now to do this. I'm trying to make kind of a reduced diversity community that does a specific function. And so I'm working a lot with cultures, and it's difficult to get microorganisms to grow together and see who kind of survives in this, and if they help each other and what the mechanism of that is. So I'm trying. It's very interesting, but it's also incredibly challenging. Yeah.
Great. Thank you all for the great questions. We will have to pause here. We will do our best to answer the rest of the questions in the chat section. So yeah, we would continue our discussion with Lauren. And to continue, how did you get into this line of research? Was it something you always wanted to do, or how did the process go about? How did you get into this?
Yeah, so I started in my bachelor's just studying environmental science and environmental biology, and everyone had kind of their organism or their group of organisms that they were interested in, and they were usually like studying lizards or mammals of some kind, or something large and charismatic. And I was never really interested in the large cuddly creatures, but I found those super weird bacteria and fungi really interesting. And it's not for everyone, but I thought it was really cool. And the fact that we didn't know very much about them was something that kind of drove me to want to figure out more.
Great. And I know even with finding a specialty, within that specialty, interest could also develop over time. So how has your work also evolved over time in terms of like with field work and the computational work you also do?
Yeah, so I like to do a little bit of everything, and I think that's clear from what I've been trying to do. So
That's good.
yeah, it keeps it interesting. So I do a bit of field work, a bit of computational work. Now I'm doing some culturing, growing microbes, and learning some microscopy. So it's a lot. Yeah, and I kind of went from doing more sequencing based work and computational work into yeah, trying to then test some of the hypotheses that we developed, or that I had thought of while doing that work, using cultures. So that's what I'm trying to do now, and tackle it from both sides.
Good. You also use a lot of computational models. How did you go into learning and working with that?
So I specifically don't use too many computational models, but I work a lot with people who are modelers.
Okay.
And so that's been a really interesting experience, discussing what needs to go into a model, and then trying to design experiments to test some of the hypotheses that are generated by the model, or trying to model things that are seen in our experiments. So it's kind of a conversation between both sides, and it leads to some really cool outcomes.
Great. So also, how does your current work look like? What are some of the things that are involved in your current work?
Yeah, so now I'm doing way too many things. So I'm doing a little bit of culturing, where I'm trying to create this group of organisms that will do specific degradation of different substrates. So looking at like chitin and cellulose specifically, which are really common carbon compounds in the soil. And we're considering using microfluidics, which is this a process where you put microbes onto a little kind of a microenvironment, and you can hook them up to a microscope and see how they grow and how they interact with each other in real time. So yeah, it's a lot of kind of developing and planning new methods and just trying things out. And yeah.
Yeah, that's great. I also know that when it comes to talking about your research, which is something you are passionate about, you can get so excited talking about it that it can even blind you from talking about some of the challenges you face doing the research you do. So
Yeah.
what are some of the challenges that you have had to deal with your research?
Yeah, so this is something I think every researcher comes across, that it's not always smooth sailing. Some days your cultures might get contaminated, or you might, I just had this happen yesterday, that my solutions weren't autoclaved properly, and so they solidified and I couldn't use them, and my whole day was kind of ruined for more or less. But yeah, so just little things like that. And sometimes you find things in your data that you didn't expect, and it takes you on a different direction than you previously thought. So it comes with good and bad things, I guess. Yeah, but failure is definitely part of it.
That's definitely true. And what are also some of the interesting and surprising results or things that you have learned doing the work you do?
Yeah, I'm very much in the process of just getting my first data set right now. So I can't say too much, I think. I don't know. Yeah, I'm just always surprised about how diverse the microorganisms are, and how they don't always react or respond in the way that you expect them to.
Yes, awesome. And you mentioned that you are still doing lots of experiments and ongoing things. And what is the future goals? What are some of the future goals for your work, if you don't mind us asking?
Yeah. This is always a tough question. So now I've been in my current postdoc position for about 2 years. And so then you start thinking, yeah, what comes next? So I'm thinking maybe I write my own proposal, try to get my own project going. I also want to include more science communication in my day-to-day work, which, because of the pandemic, really hasn't been a huge thing for me. And so that's where I'd like to go with what I'm working on.
Awesome, great. We will take another pause to take questions from our audience. And the first question is coming from Barry. And he says, I would like to know more about fungal highways for bacteria. Are bacteria proposed to transport over the surface of the extracellular matrix of the fungi, or do they have some ability to move within the fungal extracellular matrix?
So the fungal hyphae, so it's kind of like a root system, if you can imagine, for a fungus. The fungi build up kind of a water barrier around their hyphae, and this is kind of a stream that the bacteria can kind of swim. And so instead of being just like a dense kind of soil matrix where they're dependent on water coming in or out to move them around, the bacteria can now have a more direct route from one place to the other. So it's not really them getting into the fungi, but kind of using the fungi to get around. And there's some interesting papers that just came out about specific communities that associate with the fungal hyphae of specific fungi. So it's also really interesting stuff.
Good. Mandy also has a question, and the question is, are there types of soils or sediments that fluctuate more or less than any other types in their microbial diversity and composition in time, like seasonality and so forth? For example, like loam versus silt and sandy soil.
So I don't know about the composition of the soil and how much this might play a role necessarily. And people are trying to figure out where kind of the hotspots of microbial diversity would be in the soil, like globally. But when I think about an environment that fluctuates or is undergoing rapid change, I think about permafrost environments. So these are usually Arctic environments that were once glaciated and now, because of climate change, are really thawing quickly. And so this is showing us just how much we can kickstart microbial metabolism and release all this carbon. And this is a huge concern as we get deeper and deeper into the climate change struggle. So
good. The next question is making me smile, and it's from Monica. The question is, what is your favorite microbe and why?
Okay, this is really tough. Yeah, I can't say that I have an absolute favorite. Yeah, they all kind of have their little charms. Like, I think of actinobacteria, and I think it would be really cool to see them under the microscope because they grow kind of like fungi. And so I can just imagine them like sticking their little hyphae into different substrates and trying to break them down. And it would be cool to look at them and just see how that's working.
Good. Okay. Yeah, trying to be nice to all the microbes. So
I'm not playing favorites.
No favoritism. Okay. So the last question we will take is from Christine. And the question is, do soil microbes display active matter, collectively move, consume fuel, and are far from equilibrium?
So I would say yes, if I understand the question. So they're always kind of searching for new food sources, or yeah, trying to get into a kind of a optimal state where they can access an abundance of food. So yeah, I don't know exactly how to answer the question. But
Okay, good. Thank you for answering all these questions, and thanks for sending all these questions in. We will jump back into our next set of conversation, which is the last one. And yeah, also maybe we'd have some time to take some more questions. Okay, so I know you are currently calling from Vienna. Yeah, how has your work enabled you to travel and look at different ecological systems in other countries?
Yeah, so I came to Austria during my PhD for a kind of an internship. And so I think that's one really cool thing about science, that you can find scientists all over the world that are interested in the same very niche thing that you're into. And they're usually willing to let you come stay in their lab and talk about science and work on things with you. So it's been cool to meet lots of different people and see different places. Yeah, and yeah, then I ended up getting a job here, which was also, it's been an interesting experience. It's definitely, you should learn German when you come, or you should learn the language that you, that they speak in the place where you're working. So I've been working on that. And yeah, so it's been just an adjustment. And then there was a pandemic. So
Languages.
yeah.
Okay, so also how different is it doing science where you are now from your previous work experience?
Yeah, so I guess the lab structure is different. So just from like the academic standpoint. So you usually have like a one main PI who leads everything, and then a few different professors underneath who have their own research groups. And so the postdocs work closely with those professors that lead the groups, and then we're kind of responsible for working with the PhD students and the master students. And so, yeah, it's just a lot bigger and a lot more interaction between the groups than I think in a lot of the labs in the US, where you have your one professor and your one research group. So it has its advantages and disadvantages.
Yeah. I know that when people are doing their PhD research, they wonder when what next is going to be in place and when they are going to go next. So I like that you are answering these questions for us, and we are all learning how it goes. Did you think you were getting when you were getting your PhD, did you plan that you would move out of the country? Was it something you planned for initially?
Yeah, so I actually only applied to postdocs out of the US. Yeah, because I wanted to experience what science was like in a different culture, I guess. Yeah, so I kind of set my mind on that before I even finished my PhD. Yeah.
Wow, that's good. Okay. We may try that too. Okay,
It's not
so
easy, but it's definitely interesting. So
yes. Okay, so in terms of diversity in science, are there any diversity efforts you would like to highlight? And what does diversity in science look like where you currently work?
So I think I have a slide. Yeah. So I put together just a couple of the interesting like diversity and SciComm efforts that are going on here. So I think the diversity story is very much lagging behind, as is SciComm in Austria compared to other places, like specifically the US. So like, for example, even equality, like gender equality in hiring at the higher levels. Austria has, I think, 12% female faculty, which is pretty bad. So that's why this initiative called the STEM Fatale was started. And so this aims to be kind of an international platform for gender equality in the academic workplace. And so, yeah, I think this is a really nice initiative for bringing awareness to the fact that this is really a terrible ratio, and we can really do more to make this a more inclusive workplace. Yeah, in terms of other forms of diversity, the conversation is not anywhere near as open as it would be like in the US. So there's a lot of room to improve there.
Yes. So would you say things are progressing as far as representation in science in the places where you have worked? And if yes, how is that progress going?
Yeah, I would say it is getting better. People are openly talking about it and trying to reflect upon what's going wrong and what we can do better, but it's really slow progress. But I think we can get there.
Yeah.
Things will get there.
Okay. So also, how do you facilitate your science to the community you are in and to the general public?
Yeah, so here there are a few kind of outreach activities that you can do. So there's one, it's called the Kinderuni, which is where you can go to schools and you can work with kids and do kind of exercises with them about your science, and then they get to learn about what you're doing. There's also this one group called the Vienna Open Lab, which is they have lots of kind of texts and blog posts and exercises on their website that people can do, and it's in English and in German. So trying to reach a bit of a broader audience. And so I have a post up there. And then there's also what's called the microbiome support. And this is a broader European-wide initiative that works with people in clinical fields as well as farmers. And they talk about why microbes are important and kind of raise awareness about the fact that they're not all pathogens, but microbes are helpful for our ecosystems and our daily lives. So
great. So how, if you don't mind us asking, how did you know you wanted to do science communication? And yeah, how did you decide on the media to focus on?
Yeah, I'm still trying to figure this out, like which direction to go in with SciComm, but I think that it's really important to share science and get others excited about it. And I think the pandemic showed us how important that really is. Yeah, so just kind of being the voice for communicating what's actually going on and bringing about like the real facts about things. I think that's really important now more than ever.
Great. So we will be rounding up soon, and we would also take some more questions from our audience. But before that, what do you wish people knew about?
Yeah, I wish out of all this that people would get as excited about microbes as I do, just that they're really fascinating and we need them and we need to understand them better.
So that's wonderful. You're trying to turn us all into microbial ecologists.
Yeah.
Okay, so I'll take some more questions from the audience. And so this question is coming from Christine. And the question is, what type of microscopes do you use to observe and record microbes? Fluorescence, DIC, or confocal?
Yeah, most of the stuff that I've done so far is on the confocal, because you can do different types of fluorescence staining and look at where your microbes are. So, yeah.
Oh, great. Okay. And the next question is from Thompson. What type of microscopes do you use to, oh, sorry, that's what I just said. So the next question is from Karen. And the question is, do you think in the future we will know more about the optimum microbial communities that favor specific plants?
Yeah, so there's actually this is a huge area of research in Austria too. So I can speak to that they're trying to figure out which microorganisms help plants grow the best. If there's some that we can apply to plants so we don't have to use pesticides, things like that. So I think one day we'll get there, and I think we're getting closer and closer.
Good. And the next question is, can microbes sequester carbon?
So they do have storage compounds within the cells, if that's what they're referring to. So in that way, they can kind of hang on to the carbon and store it for themselves. Yeah, I think that we might have more hope for sequestering carbon with plants than with microbial communities, but I'm not sure.
Okay. And Hilary also has a question, roughly about how many individual microbes per se, cubic centimeter or averagely diverse soil. How about how many different species? Will there be a small set of common ones that dominate?
Yeah. So if we think about it in terms of like a tablespoon of soil, there are probably thousands of different species within that single tablespoon. And there is kind of this idea that similar microbes occur all around the world within soils. And so there might be slight differences within the exact species and the exact strain of the organisms, but we see kind of similar patterns.
Great. And the next question from Karen, are you aware of Microbe TV? And this week in Microbes, a great podcast. So yeah, those are great podcasts. Also from Anon, any other recommendations? Okay, so questions on some of the podcasts on microbes.
Okay, I didn't know about Microbe TV, but that sounds really cool. I did have the chance to go to the Microbe Museum in Amsterdam, which was really fascinating. So if you ever get over to the Netherlands, I would check that out. And yeah, I don't know off the top of my head any really good podcasts or anything. I think there's one hosted by the FEMS, so the Federation of European Microbiologists. They have their own podcasts. And that would be the first one that comes to mind. Yeah.
Great. And the last question is, are microbes a carbon bomb waiting to get set off? Let us know.
This is tricky too. So if we go with this idea that as the world heats up further and the climate changes, microbes will start working faster and faster and releasing more carbon, then they very well could be kind of this bomb that's waiting to release this carbon to the atmosphere. But this is still, we're still trying to understand. Yeah.
Great. That brings us to the closure of today's Life Science Cafe. Thank you so much, Dr. Lauren Alteio. It has been a wonderful session with you. And thanks to everyone for joining us. Look out for emails about our next event and that is going to be with Dr. Eve Berry. I probably screwed the name, but Evelyn Berry. And there would also be some links to the chat to sign up for the Life Science Cafe emails. And this is all part of our 10th-year anniversary. So you'll be seeing some more familiar faces and a brand new type of cafe. So we are excited to bring all of that to your doorstep. And also do well to check the Life Science Cafe website for any other information. Okay, so yeah, we would close here.